Hi, good morning, everybody. Thank you for joining today's seminar. My name's Alicia, and I'm a master's student at the IOF in the Pelagic Ecosystems Lab, and I'm studying juvenile salmon during their marine outmigration. And just to start off, first, we would like to acknowledge that the Institute for the Oceans and Fisheries is based on the UBC Point Grey campus, which is located on the traditional, ancestral, and unceded territory of the Musqueam people. You may be joining us from traditional lands across British Columbia, Canada, and the world, and we would like to acknowledge the owners and caretakers of those lands as well. I'm very excited to be introducing our presenter today, Dr. Kristi Miller. Dr. Miller received her BSc from UC Davis, MSc from UBC, and PhD from Stanford before joining Fisheries and Oceans Canada as a research scientist in 1992 and becoming an adjunct professor at UBC in 2008. She is an international leader in salmon genetics, transcriptomics, and health. Her lab developed a series of innovative high-throughput tools to monitor salmon health and condition currently applied across North America and in Norway. Since 2008, her program has applied genomic technologies to elucidate factors contributing to wild salmon declines, and she has led two multi-million-dollar multidisciplinary projects co-funded by Genome British Columbia, focused on salmon health and infection risks associated with cultured wild interactions. And she also has over 150 primary publications and has served on the editorial boards of Immunogenetics, Facets, and Coastal Marine Fisheries journals. Welcome, Dr. Miller. We're very pleased to have you. And with that being said, I'll now pass the mic on to you.
Thank you, Alicia. I really appreciate the opportunity to speak to this group today. Today, I'm going to present some of the research my lab and collaborators, many of which are from UBC, have undertaken to develop and apply novel genomic-based approaches to resolve cumulative stressor impacts in wild salmon. That's my slides. As most people in this audience are likely aware, salmon have been in decline worldwide. Moreover, the synchrony in the salmon survival trends across populations in North America suggests that there's large-scale mechanisms at play. The plots I show here are just the average marine survival plots for Chinook, Coho, and steelhead, and they show marked declines since about the 1980s and 1990s, and similar declines are present in sockeye salmon. In fact, in 2009, there was a judicial inquiry into the declines in Fraser River sockeye salmon, where many scientists testified and debated about the causative factors. The two most salient conclusions of this inquiry were that most scientists agreed that year class strength was largely determined in the early marine environment, and that multiple or cumulative or synergistic factors were at play. There was also a recognition that we knew very little about the role of infection. So understanding the factors that are contributing to marine mortality in salmon is hard, because unlike freshwater, where we can sometimes visualize changes in salmon condition and health, and if you look at the top three photos on the left here, you can visually see fish that are in distress or near dead. In the ocean, we don't have that opportunity. We don't generally observe changes in behavior, and we don't generally observe mortality events. So this makes it quite difficult to understand the factors that are contributing to mortality. When we attempt to sample juvenile salmon for our research, we tend to target large schools of fish, but compromised fish may also move to deeper or shallower waters or closer to shore, so we may actually miss them in our sampling. We also expect that cumulative stressors at play during the early marine environment, but we can measure environmental parameters and make assumptions about the tolerance limits, but we don't actually know what levels of stress will affect salmon in the field. Moreover, we may be able to detect pathogens, but how do we relate those to disease and impact? So my program, shortly after the Cohen Commission of Inquiry, I began collaborating with Dr. Brian Riddle from Pacific Salmon Foundation in the development of a new program on salmon health called the Strategic Salmon Health Initiative. And the goals of this program really were to understand the infectious disease risks that might be contributing to salmon declines, and primarily focusing on that early marine survival, which was determined to be where the majority of year class strength was being determined. Secondly, the program sought to change the face of how we study infective agents in the wild and to develop innovative and highly sensitive genomic tools to detect infection and disease in studies of wild salmon. To begin with in this program, instead of focusing on one pathogen at a time, we were interested in establishing a baseline of infection for salmon that in the coast of British Columbia that included surveillance of a diverse array of infective agents, including bacteria, viruses, protists, and fungi. And we included agents that were known or suspected to cause disease worldwide. To accomplish this, we adopted a microfluidics approach to simultaneously quantitate 47 pathogens in duplicate across 80 samples, 80 fish at once. We utilized a Fluidine biomark platform, which is a microfluidics platform, and underwent extensive evaluation of its fitness for purpose. The platform turned out to be more sensitive to detect very early stages of transmission than traditional single assay systems, but could utilize the same quantitative PCR assays that other systems used. In 3 years, we've now amassed over 30,000 fish worth of infectious agent data over 60 pathogens using this platform. To support the research on juvenile migratory salmon, starting back in 2008, we started putting out samplers on juvenile salmon vessels from DFO, covering the outmigration of BC salmon through the southern BC, sampling in the spring, summer, fall, and in some years in the winter. We also embarked on intensive sampling of smolt releases from hatcheries downstream of migrants, looking at downstream migration of wild stocks as well. After amassing about 55 papers from this program, the Strategic Salmon Health Initiative has published papers on infectious agent distributions, tracking studies associating with pathogens with migratory fate, stress challenge holding studies looking at the association with pathogens under different thermal conditions and premature mortality, infection-related risks of predation, physiological and pathological impacts of infection, disease development and pathways and mechanisms, novel salmon virus discovery, aquaculture-wild interactions. And now, for the exciting part, we've now embarked on the development of Bayesian models, which are assessing population-level associations with survival. And these span a decade of infective agent data for dozens of pathogens in Chinook, Coho, and sockeye salmon. Moreover, we've also assessed the relationships between pathogen load and fish condition, providing the most comprehensive analysis to date on the influence of infectious agents on salmon in the marine environment. So I'm going to talk about the first of the modeling papers that is now accepted in faucets that will be coming out soon. This was led by Art Bass at UBC, and it contrasts the patterns in Chinook and Coho salmon, and there will be a follow-up paper in sockeye salmon led by Amy Tecker, who was a former graduate student and postdoc of mine. Here I briefly show the culmination of the results from the first study in Chinook and Coho. This figure depicts a heat map of posterior probabilities of an association being negative for each model run with cells colored in the darker gold, showing negative associations with survival, measured as, and also salmon condition measured as relative weight, which is in the right 4 columns. Within each species, the analysis was divided up into associations in the spring and summer versus the fall and winter, respectively. So, for example, the first column is Chinook salmon survival sampled in the spring and summer period, then fall for Chinook, and so on. The pathogens depicted at the top of this plot are most consistently negatively associated with survival and condition, while those down more down towards the bottom show more species-specific seasonal patterns of impact, or they don't impact survival at all. Cells in blue are positively associated with survival or condition. We also include measures of cumulative infective risk, and you can see that if you look at the bolded titles at the right. And these cumulative risks are, This cumulative risks are strongly associated with survival in Chinook salmon, the measure of total number of pathogens, and strongly associated with survival and condition of Coho salmon in the fall and winter period. Relative infectious burden is another measure which combines the metric of infection intensity across pathogens, and that is negatively correlated with Coho survival in the fall and condition in summer and fall. Importantly, if we contrast the strength of these pathogen associations with that of the Pacific decadal oscillation, which is also shown as PDO, this is a factor that's well accepted to contribute to variations in salmon returns. And we see that for Chinook salmon, pathogens are exerting a substantially stronger impact on survival, whereas the effect of PDO is stronger than any one pathogen for Coho. Taken together, these models demonstrate that pathogenic infections are highly likely to contribute to marine survival and potentially to salmon declines. The question remains, however, will rates of infection and pathogen impacts increase with climate change, and do environmental stressors act cumulatively or synergistically in association with disease and development and survival? So I'm now going to switch gears to from a strict focus on the role of infectious agents in wild salmon to the broader need for an understanding of cumulative factors and how we're going about developing the new knowledge. When salmon migrate from freshwater natal rearing areas into the early marine environment, they're hit with a wide variety of new stressors and pathogens. While we can monitor some of the physiological parameters in the ocean, how can we determine which are actually causing stress and impacting performance, and whether these are additive or synergistically responding to each other? Again, we come back to the genomic technologies. We sought to develop a tool that would provide a more holistic assessment of salmon health and condition, combining non-lethal gill biopsy sampling, the development of targeted panels of biomarkers that could recognize and predict the presence of specific stressor states and disease states, all run simultaneously on the same microfluidics platform that we use for infectious agent surveillance. The first such panel that we developed was developed within the Strategic Salmon Health Initiative, and we developed it to recognize the presence of a viral disease state, specifically diseases caused by RNA viruses, which are the most common viruses in salmon. To develop these biomarkers, we mined microarray study data from a variety of challenge studies across a variety of different viral species and salmon species, and we identified the biomarkers that were most consistently associated with viral disease. These were tested and refined on samples from independent challenge studies. We call this the Viral Disease Development Panel, or the VDD panel. This panel can distinguish virus carriers from fish that are in a viral disease state. It can differentiate viral versus bacterial disease. It works on all tested salmon species to date. It works equally well across tissues and species, and it can be applied non-destructively to gill biopsy samples. We found that it only takes 7 biomarkers to recognize a viral disease state. So we applied the VDD panel not only to study viral disease development processes, but we also applied it to discover novel viruses in salmon. And postdoc Gideon Mordecai, who was in Curtis Suttle's lab, used this tool to discover 15 novel viruses that we did not know existed in salmon in some cases, or in salmon in British Columbia that were not part of our original panel. So this demonstrates the utility of this approach in recognizing viral disease and in studying viral disease. So we've now moved forward with the success of the VDD panel to broaden our approach to biomarker panels to a variety of stressors and physiological states potentially important to salmon survival. And these include salinity stress, thermal stress, hypoxia stress, or low oxygen stress. We also have a panel to stage smolt readiness. And obviously we have the viral disease panel. And we have a really unique panel to recognize when fish are becoming moribund. So in order to validate these panels, we conducted multi-stressor challenge studies in 2 species thus far, in Chinook salmon back in 2018, which was led by postdoc Dr. Amy Lee Hood, also at UBC. And in 2021, led by research associate Arash Akbarzada, we did the challenge studies in Coho salmon. And this involved really intensive restructuring at the Pacific Biological Station and developing this whole tank system and control system to control temperature, oxygen, and salinity over a week or 2 period. So the experimental setup included 2 to 3 smolt stages. So we actually we did these studies independently at different stages of smoltification in both of the species. In Coho salmon, we looked at only desmolts and smolts, and in Chinook salmon, we looked at pre-smolt, smolts, and desmolts. We assessed fish at all combinations of 3 salinities, 3 temperatures, and 2 oxygen levels. In the Chinook study, which was the first study, we looked at oxygen levels between 4 to 5 milligrams per liter of oxygen, and we reduced that to 3 to 4 milligrams in the Coho study because we did not see strong transcriptional responses to hypoxia in the Chinook challenge study. We had replicate tanks with 12 to 36 fish per tank, depending on the study and the size of the fish. We exposed fish for 6 days, and we sampled them if they became moribund. And at the end of 6 days, we sampled all the live fish. For Coho, we also added another component to our study. We removed the stressors and then measured stressor recovery over 3 days. So one key finding that we observed across the studies performed in both species was that full smolts were able to handle 6 days of exposure to thermal and low oxygen stress with very little to no mortality in all 3 salinity environments. So they were very robust to stress over this temporal period. But we found in both species, mortality was the highest in salt water, while brackish water was the least stressful. Overall, salinity stress relating to mortality was the stressor that was most closely associated with elevated mortality. And mortality was seen in both the pre-smolts and in the smolts, in the desmolts. In desmolts, we saw mortality in freshwater and saltwater, and in pre-smolts, most of the mortality, although not all, was present in saltwater. So this was, I'd say, a very good indication that fish may be more resilient to environmental stress as they're entering the marine environment if they are properly smolted. And this is something that we are now in discussions with hatcheries about making sure that they are releasing fish during the optimal smolt window. So in Coho, we observed a synergistic interaction between stressors. Again, the fish were resilient to thermal and hypoxia stress over 6 days, but for osmotically stressed fish, in this case desmolts, the combination of all 3 stressors resulted in 10 times higher mortality than osmotic stress plus one of the other environmental stressors. These data suggest that environmental stress is not merely additive. Alternately, in Chinook salmon, there appeared to be more of a cumulative pattern of stressor impact, with stressor levels, with similar levels of survival with osmotic stress alone or osmotic stress in combination with one of thermal or oxygen stress. But risk mortality merely doubled when all 3 stressors were present. However, in this study, we do have to consider that the oxygen levels in the hypoxia treatment were not as dramatic as in our Coho study, and it wasn't sufficient enough to resolve a consistent transcriptional response. So it's possible that contributed to any reduction in synergistic relationships between these stressors. So I'm going to talk a little bit about the imminent mortality panel. This was something that we started observing back almost a decade ago when we were doing microarray studies, holding studies in sockeye salmon where we were doing thermal challenge holding studies, and then repeat sampling and monitoring fish over time. What we found in those initial studies was when we went and looked at the data and looked at the main trajectories of the data, we expected to see thermal exposure be the main trajectory in the data. But what turned out to be the main trajectory was whether the fish were going to die within the next 3 days. And so this kind of became the fodder for our idea of developing a panel to recognize imminent mortality or morbidity. So fish, when they are becoming moribund, can visually display a loss of equilibrium. And so in our challenge studies, that's how we measured morbidity. And we sampled fish as soon as they were displaying abnormal behavior or a loss of equilibrium. And fish that no longer showed ventilation are actually declared dead. It turns out that dead fish and moribund fish display the same patterns of gene expression. And in fact, the pattern of the gene expression that is associated with morbidity actually becomes more intense, not less, in fish that die. We call this panel of gene or imminent mortality panel, and it readily differentiates fish that were live sampled in our studies and swimming from those that were losing equilibrium and sampled dead. The repeat sampling of this fish in this particular study in Chinook also showed that we could predict mortality up to 72 hours, as we had shown in a previous work on sockeye salmon. And so we can even predict mortality before fish become visual signs of morbidity. If we look at classification statistics, and here we're applying the Coho and self-organized maps. Right now, we've moved to round and forest classifiers, but for this purposes, we have a very high degree of sensitivity to detect moribund fish, with 100% sensitivity in this particular challenge trial and 82% specificity. Some of those fish that are live sampled may be at earlier stages of morbidity. We don't know, but it's still very few of them for the Chinook study. So we took a similar approach with Coho salmon. We also show a strong differentiation between imminent mortality and live sampled fish in the PCA and in the self-organized maps. Although the best separation here was when we had 3 groups as opposed to 2. And I think this will be resolved when we move to round and forest. The sensitivity of detection was still quite high at 97%, and specificity was a little bit lower. But again, it's possible that some or all of the live fish clustering with imminent mortality may have been in an earlier stage of morbidity, because morbidity was ongoing throughout the study. So we've carried out similar analyses for the other stressors, and here I show the same depictions of thermal stress in Chinook salmon. We carried out these challenges at 3 temperatures, so 10, 14, and 18 degrees. About 30% of the fish that were exposed to 14 degrees were somewhat overlapping with the fish at 10 degrees, suggesting that not all fish exposed to 14 degrees may be equally stressed. But we show that fish at 18 degrees are all showing signs of thermal stress. When we look at the classification, again, we have a high degree of specificity and sensitivity of detecting thermally stressed fish. This is a combination of 14 and 18 degrees. We have 96% specificity and 84% sensitivity, given that some of the 14-degree treatment clustered with the 10-degree treatment fish. We also observed that there's an increase in ventilation rate, in plasma lactate, in sodium potassium ATPase activity, and a decrease in glucose and growth, which are consistent with the known effects of temperature on metabolism. Again, we see similar patterns of thermal stress response in Coho. Based on only 4 biomarkers, we see a high differentiation between thermal treatments, with all of the fish at 14 degrees showing a stress response pattern in this species. If we move to hypoxia, as I said previously in our Chinook study, we did not show strong differentiation transcriptionally in Chinook salmon based on 4 to 5 milligrams per mil of oxygen compared to 8 milligrams per mil. And this analysis was done looking at all treatments together, although we did try to parse it out even further. So in our Coho study, we actually reduced it to 3 to 4 milligrams per liter oxygen to try to get a stronger hypoxia signature. And what we found was that even at this level, there was an interaction between smolt stage and salinity, whereby the optimal detection of fish under hypoxia, we had to use different panels of biomarkers. Part of the reason for this is that some of the markers for hypoxia in one smolt stage were also being expressed in the other smolt stage, regardless of oxygen levels. So this is still a work in progress, and we have a lot more work to do on this. But it may tell us that fish may be using mechanisms other than the transcriptome to deal with low oxygen conditions. Obviously, their ventilation rate and their reduction in activity may allow them to adapt. And they may be relatively resilient to short periods of oxygen deprivation. In the Coho study, as I said, we also examined how long it takes for fish to recover from a thermal stress. And this is important in interpreting any data in the field because rapid recovery would mean that you would need to capture fish in the elevated thermal environment, whereas slower recovery could indicate that exposure has occurred in the recent past. We know that from our work in Chinook salmon that we see the same thermal stress response at 2 and 6 days post-exposure. When Coho were re-exposed to 10 degrees water, it took 3 full days for them to recover to look more like fish in a 10-degree water treatment. And here, this PCA plot shows the fish that started in the 18-degree conditions in the dark red, 1 day of recovery in the brighter red. And you can see that even 1 day of recovery, they're still not looking at a similar level of stress as fish at 14 degrees, which are shown in green. But at 2 days, 18-degree recovery fish appear to overlap significantly with 14-degree. exposed fish and the first day of 14-degree recovery. By the time you get to day 2 at 14-degree recovery and day 3 at 18-degree recovery, they all intermix with the blue 10-degree fish. So basically, it takes a few days for stressor recovery at high temperatures and maybe only 1 or 2 days at lower temperatures. So this slide depicts the current state of our salmon fit chips today. And I didn't talk about some of the other panels that we utilize, but we obviously have the thermal and osmotic and still under development hypoxia panels, but we have a whole range of other panels to look at immune activation, inflammatory responses. We can also add smolt readiness. And we have pathogen assays to over 60 pathogens available, and we're starting to develop more assays to harmful algal blooms. So we can include looking at host biomarkers for stress and cDNA assays for various different pathogens and algal species. And these fit chips can be tweaked depending on what study we're doing and what kind of stressors we want to target. So I'm going to wrap up my talk by presenting some of the data from the early application of salmon fit chips. Both the data sets I'll present are applications in sockeye salmon. So the first data set is focused on stress in fresh water of the endangered Cultus lake stock. This stock is one of the most intensively studied BC stocks of sockeye salmon. And it's been in dramatic decline for over 40 years, like many stocks, but it's in such low levels now that it is considered endangered. Sockeye rear in the first year in Cultus Lake, and we know that there are elevated summer surface temperatures and there's reduced oxygen at depth in the summer and the fall. These sockeye undergo diode vertical migrations to feed, and we compared, in order to look at this study, which was all carried out on wild-caught fish, we compared the patterns that we see in Cultus Lake with patterns in a nearby lake at Chilliwack. So this just shows the measurements of the water parameters for temperature on the left and dissolved oxygen on the right during the dates that we were out there taking acoustic trawl surveys to capture fish. Note that it's important to note that when we capture fish in these surveys, we're catching fish as they're moving from the deep waters to the surface waters at night to feed. And so most of the fish are actually caught mid-water. They're not caught at high water temperature, and they're not caught in the low oxygen area. So what we find if we look at thermal stress is that, and those are shown in red, is that there are about 37% of the fish sampled in the fall and 52% of the fish sampled in the summer are clustering outside of the 95% ellipsoid for cooler temperatures. And the linear discriminant analysis to differentiate these two groups was about 89.9%. So if we look at hypoxia, and here we focused on hypoxic markers that we knew to work best in freshwater, and we focused on only the one freshwater stage, we focused only on PAR, so making it a little bit easier to not have a variety of different smolt levels. We found that there was a differentiation between normoxia and hypoxia, which showed that about 35.5% of the Cultus Lake fish sampled during months when we have hypoxia at depth cluster outside of the 95% ellipsoid for normoxia months. And there's stronger signal during the warm month period where about 50% of the fish showed hypoxic response. And again, linear discriminant analysis showed that we had about 78.3% discrimination. So importantly, we found that 91.5% of the sockeye sampled during months when both stressors were occurring, which are in July, September, and October, showed signatures of at least one of those stressors. And 25% of those sockeyes during those months showed evidence of both stressors. So this suggests that sockeye may be in a fairly strong stressor state during most of the summer months. And we can conclude that sockeye salmon appear to be impacted by thermal and hypoxia stress during this time. It's interesting to note that hypoxia stress was stronger in the summer months, even though the levels of oxygen at depth were not as low as they were in the fall. The last study I'm going to present focuses on migratory juvenile sockeye salmon, where we ran fit chips on over 2,500 fish sampled over a decade. And these were some of the same samples that we'd already run for infective agents. We're still in a very preliminary stage of analysis of this data, but there's already some important findings that I can present here. I should say that these were fish that were sampled over a trajectory from freshwater Fraser River all the way through migration in Queen Charlotte Sound. Most of the analysis I show, well, actually that's not true, never mind. So for this data set, and all data sets that we apply moving forward, we're applying the random forest classifier developed based on control samples of known stressor state. This classifier does a better job at weighting contributions of individual biomarkers to the signal differentiating stressor states. The classifier provides a numerical probability that a given stress sample is in a specific stressor state. And using our control samples, we can optimize the balance between specificity and sensitivity, and that provides the most consistent results. Once we build the classifier, we can then apply it to unknown. So in the plots here, I'm showing thermal stress on the left and the imminent mortality classifier on the right. And these are based on known stressor fish. Unfortunately, the colors aren't very bright, but the top of the panels are in purple are the fish that are in a stressor state, and the bottom of the panels in green are unstressed fish. And the vertical y-axis shows the predictor, so the probability that a fish is in a stressor state. We can actually, if we want to classify fish as positive or negative for a stressor, we can look at the sensitivity and the specificity under different thresholds. We find here that about a threshold of about 70% probability provides a very good balance of sensitivity and specificity to predict stressor states, and is somewhat conservative to ensure that we don't predict the presence of a stress when as a false positive. So when we apply this to our wild migrating juvenile sockeye salmon, what we get from the classifiers is a series of these predictor scores. And here I'm just showing an example of a small number of fish, showing what those predictor scores look like. And I'm highlighting the ones in red as those being a high predictor that particular stressor is present, showing thermal, osmotic, imminent mortality, and the VDD panel stressors. And I have it sorted for imminent mortality, which is why there's so many of those. So if we look at the predictor values for thermal stress as a continuous metric rather than as a threshold response, the plot above shows the thermal predictor on the y-axis and the distance from the Fraser mouth on the x-axis. This came from work that Art Bass has done, and he's doing a lot of the analysis of this data set. The points were fit into a simple generalized additive model or a GAM that are shown in the orange curve. The blue side of the plots is samples taken from freshwater, where you can see a rise in thermal stress occurring at the Fraser mouth, and a sharp rise as the fish migrate through the Strait of Georgia. As fish enter the Johnson Strait, which is a much less stratified and mixed water, much less stratified and contains mixed water from the West Coast, the temperatures in that area do decline. And the plot at the bottom, the map at the bottom, basically shows the thermal profile for 1 year over the time period that we would be sampling these fish in the early or the late spring, early summer. And you can see that the Strait of Georgia, shown in yellow, is where we see the highest levels of sea surface temperature rise. So here's what those annual deviations look like for our study from years 2008 to 2018. And the x-axis is year, and the y-axis is the model coefficient for each year, which represents the deviation from mean across the years. The red line would be the mean, so 0 deviation. The size of the points reflects the number of fish we had for each year. So we have a lot more fish in some years than others. And that would somewhat reflect, you know, your the meaning or the meaningfulness of the measures. Interestingly, we see a steady climb in the deviations across the decade, suggesting that thermal stress is increasing annually. To determine if this was truly related to sea surface temperature, we gathered spring and summer time, basically April through August temperature in the Strait of Georgia region, overlapping with the points from our analysis. So this came from NASA, NIRS, and SST. The plots show a regression, and the regression is quite high at 0.59 between the average annual sea surface temperature and thermal stress annual deviation, with the points labeled by year and scaled by sample size again. Clearly, there's a positive association with sea surface temperature and thermal stress. We don't yet know what this means in terms of survival impact. That will be some of the next work that we focus on. So very briefly, this plot shows similar patterns, or shows the patterns of osmotic stress across the same study area with model predictions based on 2 stocks in the Fraser River sockeye salmon that were tracked from freshwater through the early marine residents. And we see the highest rates of osmotic stress in freshwater, which is to the left of the red vertical line, and a second peak that occurs around Johnson Strait, which is where fish are exposed to the highest sea surface salinity levels. There are a few things that we can conclude from the preliminary analysis of our sockeye data set. First, that thermal stress is the dominant stressor observed, peaking at 57% of the fish sampled in the northern Strait of Georgia over all years combined. This is consistent with the variations in sea surface temperature. Osmotic stress is highly correlated with imminent mortality across seasons and years, similar to findings from laboratory challenge studies. And osmotic stress and imminent mortality, which I didn't show, are highest as fish move from natal rearing areas to the ocean, so in the freshwater environment. And this is consistent with the daily mortality rates from tracking studies. So we are going to be very interested in employing our imminent mortality data with models looking at variations in survival in different environments. So the salmon fit chips are being applied in the same microfluidics approach as our infectious agent monitoring. And we have multi-stressor talent studies that we've carried out in 2 species, and we'll be carrying them out in more. So in terms of the osmotic stress signal, it's important to note that smolt readiness is not the only potential factor that will come into play with osmotic stress. But we also have the potential of osmotic stress coming about from infection in gill or kidney tissue. We have many agents associated with poor survival in juvenile salmon that are known to infect these tissues. But it's also important in terms of wounding and infection in skin. So intense levels of sea lice infection, lamprey predator bites, bacterial infections from Tenacibaculum, Mortella, and Yersinia ruckeri, and fungal skin infections like Ichthyoboda can all result in osmotic stress in fish. And so what we don't know yet when we're sampling fish in the marine environment and we're seeing evidence of osmotic stress is whether that osmotic stress is coming from something from the freshwater environment, or it may be due to wounds or other infections in the marine environment. And that's something we're going to be focusing on. So now we're seeking solutions. We need to know a lot of things to mitigate. So the whole purpose of our program was to better understand the stressors and diseases that might be undermining salmon performance, especially in the early marine environment, but to use that information to identify what we could possibly mitigate and what mitigation would look like. Obviously, mitigation might be easier if there was a synergistic or multiplicative response to stress, where if you remove one stressor, you would considerably reduce mortality. But it is also possible in some cases that stress is cumulative or even antagonistic. So where the key, the questions I think we still have to ask is, what are the key early marine habitats used by stocks of concern? We know that a lot about that for Fraser River stocks, much less in terms of some of the West Coast stocks or Northern BC stocks. What are the stressors or diseases impacting survival, and where do they pose the highest risk? That's something that the fit chips are going to be looking at. How are key infections transmitted, and when is disease manifesting? Are there synergistic relationships between stressors and diseases? And what stressors or diseases are amenable to mitigation? And these are going to largely be ones that are under anthropogenic control and have anthropogenic triggers. And then what would mitigation look like? And, in my view, with overall rates of survival plummeting to less than 1% for some stocks in some years, if we could even increase survival by 2% in the marine environment, we could make the difference between sustained populations and the continuous decline towards extinction. So lastly, I just want to recognize that this is a huge body of work that I'm presenting today that's involved a lot of people, many people from UBC, both professors from UBC as well as graduate students and postdocs. I want to thank all of them. And I also want to thank my funders, Genome, British Columbia, Pacific Salmon Foundation, Fisheries and Oceans, NSERC, and also our partnership with Gunther Analytics, who's performed a lot of the bioinformatics work for us. And that is the end of my talk.
Thank you, Kristi. That was incredibly interesting and super comprehensive. So we are now at the question and answer period. So I see that Paige Ackerman has posted a question in the chat box if you want to read it, but I'm going to encourage Paige to also ask the question.
Okay, I have a voice now. Yeah, Kristi, really interesting talk as always, thanks. You know that I have an interest in BKD, and so my question is basically, we know that anything that targets the kidney, it also affects renal function, and BKD is a really good example of that. So I'm curious that when you were looking at the signatures for salinity tolerance and smolt readiness, did you correlate that at all with any of the pathogenic profiles, in particular Renibacterium? Because I can see that being a really interesting application for pre-release disease management, both for aquaculture and for enhancement, that might get overlooked through traditional pre-release or pre-transfer screening practices, and could actually provide an opportunity for therapeutic treatment prior to release.
Really, really good question. And the data that we have will allow us to do that in our wild migratory fish. All of the challenge fish were tested before the challenge for the full panel of disease agents that we look at. And so they didn't have, fortunately, any Renibacterium in our challenges, which I know can be a problem in challenge work. But yes, that is the intent moving forward, is to be able to look at the interaction between infection and these different stressor states that we observe in fish. So absolutely the next step.
Cool, thanks very much. I look forward to seeing that work done.
Thanks.
Thank you, Paige. Brian, you've got a question. Go ahead, please.
Yeah, thanks, Kristi. That was a fantastic talk, as usual. An amazing body of work. And I wanted to ask if you've done any exploration of the interaction between nutritional health and stress responses.
Well, that turns out to be one of the next challenge studies that we intend to do, and we just got animal care approval to do that. It was a little hard in coming. So I think that is going to be really important, and you and I have already talked about that a little bit. We will be in one of the next challenge studies, we will be looking at the combination of temperature and food deprivation. One of the goals of that is going to be, understanding how food deprivation affects our thermal response, but also in determining whether or not we can identify biomarkers to detect nutritional deprivation in gill tissue, which may not be possible, but we are going to try. I mean, gill tissue is great for a lot of things, but not necessarily metabolism and energy and feeding. But we are going to be looking at that.
That'll be fantastic. I just want to ask a quick follow-up. Will you be focusing on quantity or quality of prey or for food, or both?
In this particular study, it's going to be quantity. It's not as fancily designed as looking at quality. But if we are able to see much signal with quantity, that would be a good follow-up question. I mean, certainly quality of food. I mean, those kind of studies have been done a lot in terms of work on aquaculture and hatcheries and stuff. And it may be possible to be able to marry some of our approaches with samples that are taken, during some of those studies, so that we don't have to repeat everything. But most people do not take gill samples when they're looking at diet. So that's probably not going to be commonly available.
Thank you.
Thank you, Kristi. Gabrielle has another question for you. Go ahead, Gabrielle.
Thank you, Kristi, for this super nice talk. I have a small question. All your work are based at the stock level or a part of the distribution of the species level. And I was wondering do you think if you were working with the full distribution of the species, and so with all the stocks, do you think you will have the same result for the stress level analysis? And because you will expect to have a wider thermal tolerance?
That's absolutely true. We do expect, I mean, there's some really lovely work done out of Tony Farrell's lab by Erica Leisen that has shown that there are differences in the level of stress that even the different Fraser River sockeye salmon stocks feel, with Chilco requiring a higher temperature to actually feel the effects of thermal stress, and other stocks being more sensitive to lower levels of increased temperature. So yeah, we do. I think that it's going to change dramatically the kinds of biomarkers that we use to recognize those stressors? Not too much. You know, so I didn't really show this kind of data, but for thermal stress, especially, and really imminent mortality as well, there's just a huge overlap in the biomarkers that we see associated with both of those stressors. There's a little bit of more difference in osmotic stress and in basically smolt stage between species. So there is more work that needs to be done there. We this year are going to be carrying out the same multi-stressor challenge studies in chum, pink, and sockeye salmon. We've already done a little bit of work in sockeye, but we're going to be doing a much more thorough examination. We are actually applying this approach in sea trout in Norway as well, and in Atlantic salmon in Norway and on the east coast of Canada. So, you know, we're pushing the limits a little bit, going out into more and more species. Obviously, the approach is going to work the very best when we've got control studies for within a species. We'll never have control studies within every stock because that's just simply impossible. But yeah, I do think that you know, what you see as the level that triggers a specific stressor response may vary between different species. And that's one of the jobs of the multi-stressor challenge studies, and contrasting them and trying to keep our levels that we're exposing them to similar, is to be able to look at that. Like, for example, you know, the Coho at 14 degrees all showed, you know, a significant stress response, whereas in Chinook, about 70% of them did. So that also kind of shows the difference in stressor resilience for thermal stress between the species.
Thank you.
Thank you, Kristi. I don't see any other questions for now, so I'll squeeze in a little one whilst other people think about theirs. I had a very specific question. I was just curious as to why you picked 6 days in some of the initial trials that you had.
Well, we were looking to develop panels that would recognize chronic as opposed to acute stress. Chronic stressors are more likely to have a survivorship impact, although that's not always the case. And it's not uncommon. I mean, if you look at, for example, we've done a lot of work on return migrating Fraser sockeye salmon that have to migrate through the mainstem Fraser River. And what we find is that they're oftentimes exposed to, you know, high levels of thermal stress in the mainstem Fraser River for, you know, up to a week period of time. And if they get really stressed, they oftentimes will move off and go into small, cooler streams or dive deep into the lakes to reduce their level of stress. And in many cases, we find that fish that have higher levels of infection will undergo that behavior more than fish with low levels of infection. So we know that, we also know from our work in sockeye salmon that 6 days of thermal stress in an adult sockeye salmon is enough to kill them in some cases. Now, we didn't want to use mortality as our endpoint, but and in the first study, we looked at 2 days versus 6 days. So we did sample fish as well at 2 days. So we wanted to sample them long enough that if there was going to be a survival consequence, that we would be able to at least measure the beginnings of that, but not so long that we ended up killing all of our fish.
Thanks for that answer. Brian, you had a follow-up question?
Yeah, so like in the marine phase where you're looking at, you showed some satellite maps of, you had SST data from that light. So surface temperature, and the fish are obviously not, you know, right at that surface layer. So I'm wondering how well you think the measured environmental data relate to the actual experience of the fish. And how can we improve our understanding of what is actually experienced by the fish in the future?
That's an excellent question. I should have brought that up before, because that actually is what I think is one of the more compelling aspects of what we learn. Because in the ocean, fish can move away from high water temperature. They can move away from surface layers and dive deeper into the ocean. And so you could say, well, why would they experience thermal stress when they can avoid it? But the fact of the matter is that the actual data of fish in the field suggests that they are not necessarily avoiding it at all times. And you know, most of these species do go to the surface layers to feed. They could go at night. Oftentimes that's what they do. We do have information about the depth that we capture fish, because in all of our cruises that we use to collect our fish, we have information of depth of capture. We do know, again, the studies that I showed were sockeye, and sockeye do tend to be more surface oriented at that stage in their life history. But you're absolutely right. You know, the question is, and you look at the cultist fish, like, why aren't they using the middle of the water column where there's no oxygen deprivation and there's no high water temperatures? The fact of the matter is that because we can see those stressor responses, they're clearly not completely avoiding those environments. And the fact that we saw when we remove stressors, that it takes some period of time for them to become non-stressed, means that when we're seeing the stress, it doesn't mean that those fish had to be in the surface waters for days and days. They might have been there and they might be going up and down, but they're still stressed by it. That they keep that stress. If the temperature is high enough, if they're in a high water temperature condition for long enough, that stress is going to stay with them for a long period of time. And what that means is we do have to be concerned about that when it comes to, you know, climate change and what our expectations are in how that's going to affect salmon. What I didn't show was that hypoxia stretch, which I recognize has been a real difficult one for us to get a handle on. If we remove the oxygen stress, there's zero signal left within a day. So they seem to, A, they don't respond to it as strongly, at least using the biomarkers that we've used, but B, they seem to be able to come back from it very, very quickly.
Thank you, Kristi. That was a great answer.
I don't see any other questions, Kristi. So, Paige has another question. So go ahead, Paige, and then Alicia, you can wrap it up after that.
Okay, sorry, just one follow-up one. And Kristi, you might have covered this and I might have just missed it. When you were looking at the pathogenic signatures, are you getting just a presence-absence of any given pathogen, or are you able to detect a magnitude of presence of a given pathogen?
It's quantitative, so it's qPCR. All of our data gives us a CT value and we calculate copy numbers. So in the models looking at survival, we simply use presence absence. So we use the proportion of fish in a given year that contain that particular infective agent. But in our models that are looking at fish condition, which is we used a measure of relative weight, it was quantitative. So that measure was looking at how the intensity of infection affected the condition of the fish. So survival was based on presence, absence, or prevalence, and condition was based on intensity.
So just to follow that up, could you be using this technology then to do some early sampling, say broodstock sampling, non-lethal on a conservation stock, to potentially do some BKD screening, where perhaps we might not have to cull eggs at a certain level because of handling or holding risk?
The short answer is yes. You know, to use non-lethal sampling, we first, you know, have to establish that we can detect at a pretty good level of sensitivity a given pathogen. If you're talking about not culling fish, but absolutely, that is one of our goals, is to be able to provide tools that hatchery facilities can use to be able to monitor the condition of fish. One thing I didn't talk about in this talk today, the other area that we're working in is in the area of environmental DNA. And environmental DNA, you know, where you're measuring the composition of species within a water mass using filtered water samples. We also do that with infectious agents, and we've done quite a lot of work on that. And so there is a potential that in a hatchery environment, we could simply measure the levels of Renibacterium within the tanks to get an inkling of what tanks you may have to be worried about when it comes to the levels of that pathogen. So we're ground truthing this right now with 10 different aquaculture farms sampled over every month for a year, where we're sampling the fish and we're sampling the water in the pens and around the farms. And then we're trying to match the signatures that we see of the infective agents in the water column with the infection levels of the fish on the farm. We're also doing this in aquaculture hatcheries.
Okay, thanks.
Thank you, Kristi. Yeah, just before wrapping up, as it is almost 12 o'clock, I would just like to take a moment to thank Dr. Miller on behalf of UBC's Institute for the Oceans and Fisheries. And as someone who works with juvenile salmon, it was very interesting to hear your work about these technologies, determining fish stress and mortality. And I'm also very excited to hear about your future projects looking at stressors relating to nutritional condition as well.